Okay, looks like there's quite a good number of you in here now, so I'll get started. And just want to say a huge thank you for everyone for joining our final talk now of Careers Week. Today, we're very lucky to have Professor Robin May sharing his career so far and some tips and tricks he's learned along the way. But before we kick things off, I just want to share a few slides to you guys to introduce Protein Tech in case you are unfamiliar with who we are. So Protein Tech is a leading life science reagent manufacturer of antibodies and immunoassays. We have an extensive portfolio of antibodies that encompass many different research areas. We also offer kits such as ELISA kits and IHC kits, as well as our cell separation kits and antibody labeling kits. Within our Protein Tech brand, we also have our humankind branded cytokines and growth factors, and all of our recombinant proteins are raised in HEC293 cells, so you can be confident that they have the native folding, post-translational modifications, and glycosylation that you'd expect to see in human proteins, which ultimately increases their bioactivity. We also have our Chromotech branded nanobody-based reagents, and nanobodies are about 10 times smaller than traditional antibodies and combine to their targets with really high affinity, making them ideal for immunoprecipitation, super resolution microscopy, and protein purification. And lastly, then, we have a subsidiary of Protein Tech called Protein Tech Genomics that manufactures antibodies conjugated to oligonucleotides to allow for protein detection alongside your single-cell RNA sequencing. All of our award-winning products are manufactured in-house, and we have the largest portfolio of antibodies for the human proteome, as well as a rapidly growing recombinant portfolio. We truly understand the importance of high-quality validation data, and all of our validation data is open access to all of our customers, and we try and perform knockout and knockdown validation wherever possible. And as antibody experts, we have in-depth knowledge of immunoassay applications, and we can offer outstanding technical support to all of our customers. And we have 7 inventory sites with manufacturing and research across 3 different continents, allowing for global next-day delivery. Finally, I just want to say that we define our success as a company based on your scientific success. We have over 300,000 citations globally, which puts us in the top 5 cited antibody companies. And then I just want to highlight that as a company, we strive to make the searchability of our products as user-friendly and informative as possible. And we've actually got a new website update that we've launched to help our customers select the right antibody. So this includes our unique 3D epitope viewer that allows you to interact with epitope structures and explore antibody binding sites so that you know exactly where our antibodies will bind to your protein of interest. We've also introduced advanced filter options to help you find exactly what you need, such as our immunogen region selector toggle that allows you to filter for antibodies generated from your region of interest. And additionally, you no longer have to filter through all of the variations of an antibody from the search results. You can view all formats, including conjugations, buffer formulations on just one page, which ultimately will streamline the process of antibody selection. But that's everything I wanted to say about Protein Tech. I'd now just like to introduce our speaker for today. So Professor Robin May took up his role as the Chief Scientific Advisor for the Food Standards Agency in July of 2020. In this role, he provides expert scientific advice to the UK government and plays a critical role in helping to understand how scientific developments will shape the work of the FSA, as well as the strategic implications of any possible changes. He's responsible for the integrity of science and evidence that underpins FSA activities, as well as for communicating the FSA's evidence-based approach to other government departments. So Professor May's early training was in plant sciences at the University of Oxford, followed by a PhD on mammalian cell biology at University College London and the University of Birmingham. Then after postdoctoral research on gene silencing at the Hubrecht Laboratory in the Netherlands, he returned to the UK to establish a research programme on human infectious diseases, and he worked as the director of the Institute of Microbiology and Infection at the University of Birmingham. So as well as continuing his work on infectious disease at the University of Birmingham, Professor May is a fellow of the American Academy of Microbiology and the Academy of Medical Sciences and specializes in research into human infectious diseases with a particular focus on how pathogens survive and replicate within host organisms. In May 2022, he was appointed as the 39th Gresham Professor of Physics, a role in which he provides public lectures on medicine, health, and related sciences. And with that, I just want to say a huge thank you to you, Robin, for coming to speak to everyone today, and I will stop sharing my screen and let you take over.
Brilliant. Thank you so much, Bobby. I hope you can hear me. You made me feel very old with all of that, but happily you've chosen a nice young-looking photo of me, so that makes me feel much younger all at once. Brilliant. Thanks so much for having me here. It's a real pleasure to sort of see you all online, even though I can't actually see you. Good afternoon for those of you in Europe, morning for those of you in the US, and evening for those of you everywhere else. Nice to have everybody here. I'm going to try and share my slides now, so we might fall at the first hurdle, but let's find out. There we go. And then I'm going to, oops, do that one and that one. And then I'm going to rely on you probably to tell me if that's working.
Oh, I can't quite see anything just yet. So like,
Oh. Shall I stop?
oh, there we go. Sorry. Yeah, absolutely working fine. Thank you.
Brilliant. Right. Well, and if I suddenly disappear offline at some point, someone will have to come on and say, you've stopped moving your slides or you've gone awfully quiet. Or if I completely disappear, just talk amongst yourselves and make jokes. Um, uh, Great. It's a real pleasure to be here. Thanks so much for taking the time and for organizing this, actually. I spend quite a lot of my time talking to people about kind of careers in science, academic science, government science, industrial science. But also, I'm just really very passionate about people kind of knowing what's out there and feeling kind of free to do different things and to move between disciplines. So it's great to see also some familiar faces who've done that here on the call as well. So what I thought we'd do over the next sort of 30 or 40 minutes or so is kind of put together a few things. So I'm going to do a little bit of biography. I always feel really sort of, well, A, old and B, immodest talking about biography. So I'm going to try and keep that quite short, but I sometimes think it's quite useful to hear from other people about how they got to where they got to and what they did along the way, and maybe also where it didn't quite work as it should have done. I'm going to talk a little bit about my role as Chief Scientific Advisor, but also the kind of general science advice system. For those of you joining from outside of the UK, this is obviously different in different countries, although actually in some senses it's quite comparable with many countries that have that. And in fact, one of the pleasures of the role is that I'm on the international group of science advisors that's meeting in fact next week. And so most countries now have Chief Scientific Advisors who have kind of similar roles. So hopefully it will apply also wherever you're listening in from. I'm going to talk a little bit about my current role, but most of all, I'm really keen to make sure we leave plenty of time for questions, because the most interesting bit of these kind of things, at least as far as I'm concerned, is hearing your questions and comments and thoughts on all of this. So I'll try and make sure we have plenty of time for that at the end. Right, so where to start? So I guess people often say to me, what kind of scientist are you? And I've sort of varied over the years in saying I'm kind of a geneticist and a microbiologist and an immunologist. And these days I just say I'm a biologist. And that I think is reasonably fair. I've done quite a variety of different biological things, but I've always been really interested in biology. And in fact, you can see a piece of evidence here. This is me doing a bit of biologizing, aged, I think, about 2.5 with my grandparents' dog. I can't actually remember if I was thinking biological thoughts at that stage, but definitely I do remember being very, very young and being interested in insects and plants and soil and all those kind of things. But the thing that probably most inspired me to kind of do biology as a proper career, and I suspect I am not alone in this, I'll be interested to hear afterwards who else shares this one, but the single person who's probably most influential in making that decision is this person here, David Attenborough. And for those of us of a certain vintage, i.e. me, we kind of I grew up on a childhood diet of just what felt like endless David Attenborough documentaries, all of which were absolutely fascinating and eye-opening. And the one that really, really inspired me was this one called Life on Earth. I strongly recommend. It looks a bit grainy now if you look it up, but it is still a great listen and watch. And I have very clear memories of David Attenborough sort of disappearing around the world, looking at both fossils and live organisms and kind of reflecting on the evolution of life on Earth. And I thought, to be frank, I thought that is the job I want. I want to be trekking around the world on the back of a donkey and making great television. Still haven't achieved any of those aims, but maybe this time yet. But definitely that kind of diet of sort of science documentaries got me hooked on thinking about becoming a biologist. And actually, the sort of biologist I thought I would become was actually a vet. I was quite keen on becoming a vet. Vet in the UK sense as a veterinary animal clinician, not a veteran, military veteran, I hasten to add. So I was kind of all set to become a vet. And when I was at school, I did all my subject choices to go and do veterinary science. And in fact, very close to the deadline for applying to university, I suddenly decided that I probably didn't want to be a vet. And actually, I was more interested in kind of general biology. And so I was looking for a degree that was kind of very general biology. And I actually ended up, fortunately for me, in Oxford, because they at the time ran and still do run a very general biological sciences degree program in which you are forced in your first year to do lots of different topics. And at the time, I guess I was slightly like many 18-year-olds, slightly thinking, why am I having to do these subjects I don't really like, I'm not interested in? But actually, with the benefit of hindsight, that was a real lifesaver because nowadays I use some of those things I learned in those compulsory modules at school on a kind of daily basis. So I went to Oxford, I went to St Anne's College, which is the picture in the middle. I have to say I lived in a bit of it, which is a lot less beautiful than that, but it was a great experience. And whilst I was there, I think that is where I first got kind of hooked on this idea of pathogens and infectious disease. And in particular, something that I've spent a lot of my life doing, which is staring down microscopes. And there are two people in particular who were just fundamentally influential in that decision. And so I want to kind of name check them here. So one of them was Sarah Gurr, now Professor Sarah Gurr down at Exeter University. And Sarah ran this module when I was undergraduate called was sort of pathogens and parasites. That's what it's called. And what was really unique to me at that time was this module did not think about human disease or animal disease or plant disease. It thought about all of disease, and it spent a lot of time making comparisons between pathogens, for example, on bananas and on skin. And that was really influential to me in thinking about the way we think about diseases, and in particular in thinking about infections not as a problem to be solved, but as a kind of part of the wider evolutionary ecosystem, and therefore something that if you really want to tackle them, you have to think of in a particularly sort of evolutionary way, rather than just a let's kill everything kind of way. So Sarah's module was particularly influential, and actually, I'm still in touch with Sarah, and she has been very influential throughout the rest of my career. Which is probably lesson number one, I would say, is you'll pick up along your careers these people who are great mentors and hang on to them for everything they're worth. Because I still rely on advice from mentors who I've been talking to for 30 years. So a good mentor is worth their weight in gold. So that kind of module, Sarah, got me interested in this idea of pathogenesis and infections. And at the same time, I did my project. I guess most of you will have done science degrees. And when you do a science degree, in most places, you do some kind of research project. And the research project I did was with this guy here, David Shotton, and it was focused around microscopy. It was actually focused primarily around microscopy of white blood cells, and it kind of got me first interested in this idea of chemotaxis and motility in white blood cells. But rather more fundamentally, it introduced me to this kind of amazing world of microscopes. And sadly, I don't have a picture of it. I wish I did have a picture because you would be able to laugh your socks off. So the microscope I used at the time was state of the art. I was very fortunate, state of the art advanced time-lapse microscope. And it was about the size of a garden shed. And it recorded its very advanced time-lapse images onto Betamax video, which is something that most of you on this call will never have heard of. But long before we had Netflix, we had DVDs, and before DVDs, we had video cassettes. And right at the beginning of video cassettes, there were two rival types, VHS and Betamax. Betamax died a death, but you can date the time I was at Oxford because their period it was still a viable format. And so tucked away up in my attic, I have some completely unplayable Betamax videos from that period of time. But notwithstanding the technology, it was a really eye-opening moment for me, into the fact basically the cells move. So everything else I'd done had been kind of stationary, but now I was making movies. Here's a little screenshot of one of those very advanced movies from those time. You can see the nice annotation that I did there, where we're mapping out individual cells moving. And it got me really, really interested in this idea of studying infection through time-lapse microscopy. So I finished my degree at Oxford, and you'll notice a bit of a theme arising here, because having decided I was going to be a vet and then changing my mind at the last minute, I'd gone to Oxford, I'd done general biology, and in fact, because I'd done largely plant-based modules, I officially qualified as a plant scientist, which was great, except for the fact that at that stage of my career, I decided I didn't really like plants very much. But I did like science, I did like microscopes and research, and so I was keen to do a PhD, but not really in my specialist area at all. So I was looking for a PhD that would allow me to try out different areas. And that, of course, is now a very common thing in doctoral training programs. And Poppy, our host, is a great example of that, where you do different projects. But back in those days, that was completely uncommon. And in fact, there were only two programs in the UK when I was applying that offered this opportunity to do rotation projects, one in Edinburgh and one in London. And so I ended up in London at UCL. And in this program where you did multiple projects in your first year and then chose one for your PhD. And that was an absolute godsend. And it was a godsend for two reasons. One is that I obviously didn't have very much skill in this area at all. I basically spent most of my undergraduate doing sort of plant biology. And now I was in a department where everyone worked on animal cell biology and biochemistry. And so I had to do some kind of speed learning. So it was really good to be able to kind of rotate around projects and not kind of waste the first year of my PhD having to learn stuff. And the second reason was it's a great lesson for me in expectation management, because I went to London completely convinced that what I was going to work on was HIV. This was before we had a good therapy for HIV. I was really interested. It was a major, major issue, obviously, across the world. I was super keen to work on viruses and HIV, and I was so sure I was going to do that. And indeed, one of my projects in my first year was on HIV, and I absolutely hated it. I really, really didn't like the project. It was not I was not interested. I couldn't do it. And if that had been my kind of full 3 or 4 year PhD, I suspect I wouldn't have finished. But because I was on a rotation project, I could do different projects. And actually, one of the ironies is the project that I chose almost as a kind of last choice, one extra, was with this lady here, Laura Macheski. And it turned out to be just fantastic. The project was really interesting despite my expectations. We made good progress in the kind of 3 months that I was doing it. And most importantly, Laura was just the best type of supervisor you could possibly imagine. And I do think, I'm sure many of you who've done PhDs will say the same thing. Your supervisor is kind of make or break. And Laura was absolutely mate. She was incredibly supportive, really enthusiastic, had a endless selection of mildly inappropriate jokes, and it was just a great way to do a PhD. So I decided to choose my PhD with Laura. Actually, one of the other things that happened at the time was, whilst I was rotating with Laura, she let me know that she was actually thinking of moving the lab. And it was very interesting. I got a lot of advice from people saying you should never join a lab that's going to move. You'll waste your PhD packing boxes and moving boxes. And actually, nothing could be further from the truth. That lab move was a really, really useful bit of learning for me. When I set up my own lab years later, I could kind of draw on the experience of setting up a new lab. And actually, it didn't delay us really at all. And so I guess, encourage people to take with a pinch of salt some of those kind of pieces of advice you might get from people who say you must not do this or you must definitely do that. Think for yourself and take a chance sometimes. So with Laura, I started my PhD. The lab, as I said, moved halfway through my PhD, but with Laura, we worked firstly on a problem which actually still sticks with me, which is the problem of Listeria monocytogenes. This is bacteria, as you may know. It's a cause of food poisoning, particularly from things like soft cheeses. For most of us, it's not really a problem, but if you're immunocompromised, or in particular, if you're pregnant, this is very serious because it's a bacteria that can cross the placenta and abort your fetus. And so there's a lot of interest in how this bacteria spreads. And in particular, one of the things we knew at the time was that this bacteria swims inside your own cells, inside human cells. And it swims like this. Hopefully you can see that moving on the screen in front of you. So it swims actually by hijacking host cell proteins. So the bacteria itself is not motile, but it borrows machinery from the mammalian cells in to drive its own swimming process. And so one of the things we were interested in is, if you could block this swimming, you might be able to block the spread of the bacteria. And so working with Laura, and actually with a collaborating group in Germany, we had a careful look and managed to work out that there was a single protein of the host cell that was really critical for the swimming process. And still, one of my favorite experiments I've ever done was this one, when we put the same bacteria into the dish and we blocked that protein with an antibody, not a protein tech antibody, I'm afraid, but nonetheless an antibody. And what you can hopefully see just about on that right-hand movie is that these bacteria jiggle around on the spot, but they don't swim anymore. And so that was, I guess, my first kind of real insight into sort of molecular pathogenesis, which has been a bit of a theme ever since. Um, so so did this PhD with Laura. We worked on Listeria and then went on to work a bit on macrophages actually, and chemotaxis and phagocytosis, which is something I've kind of picked up on much later in my career. Then I finished my PhD, and I was fortunate enough to get a fellowship from the Human Frontier Science Programme, which another shout out there. That is a great funder. If any of you are listening to this thinking about applying for postdoc fellowships, I would super strongly encourage it. Really supportive organization, flexible money, lots of benefits, really, really good. And with that fellowship, I was able to move countries, and I chose to go to a lab in Utrecht in the Netherlands, which is this picture in front of you here, to work with the person you see at the top of the screen, Ronald Plastic. And this was a bit of a jump into the unknown because Ronald had a big lab actually working primarily on genetics, and in particular at that time, and you can again test the vintage of this story, because at that time the really big news that Ronald's lab were working on was this new phenomenon of RNA interference. And RNA interference, obviously, as you know, is a way of silencing genes. And at the time, all we knew was that it worked. No one knew how. And I was really keen to develop some new skills. In particular, I was really keen to work on a model organism. I'd previously only worked on cells, basically. I'd never worked on a whole organism. And Ronald's lab in particular was famous for working on this guy, the nematode worm C. elegans. And so I thought I could go off to the Netherlands and I would learn a bit about this model organism. I'd be in this kind of cool and exciting environment of RNA interference, and it would be a great learning curve. And most of these things were true, but there were sort of two things I hadn't really calculated in. The first is that Ronald's lab was, I mean, it was absolutely cutting edge. He was, fellow of the National Academy and incredibly pioneering, but it was huge. It was about 35 people or something when I joined. And I came from a really small lab of about 3 or 4 people. And I have to say, it took me quite a long time to adjust to that sense of scale and that change in scale. And I guess that's something that has sort of stuck with me now, that when I've changed jobs since, that you've got to kind of think about these things that are not necessarily in the job description, like who's there and how many people and these kind of things, because those are big factors in your kind of day-to-day enjoyment too. The other thing that I really had not reckoned with, and that I should have seen coming, is that this was a huge discovery, and it of course went on to win people Nobel Prizes. And so the field exploded in the 3 years that I was in Ronald's lab, in such a way that there was absolutely no way I was ever going to find myself a faculty position working on this, because the rest of the world was looking for faculty positions looking on this, many of whom were much better experimentalists than I was. So about midway through my postdoc in Ronald's lab, I decided, okay, this is probably not what I'm going to do for the rest of my life. I need to think about doing something different. And actually, I had a particularly gloomy day, I remember, gloomy week, in fact. It was a Friday evening. Literally nothing had worked. I'm sure many of you on this call know that feeling. Every experiment had gone wrong. You just knew there was no kind of future in this project. You knew there was not a job at the end. You thought, what am I doing with my life? And I tell this this story to my colleagues now. And so I thought in the evening, well, I've got to do something else. I've got to get out of academia and research and do something else. What can I do? I know what I can do. I can join the civil service, because they like scientists. They need a lot of scientists. And at the time, there was a web-based quiz that was run by the UK civil service to assess whether you were suitable for a career in the civil service. And so I sat there on this Friday evening, cup of coffee in one hand, filling in endless questions about, my kind of personality and my interests. Press sort of submit at the end, and about 5 minutes later, got a little email back automatically from the system saying, "Thank you for filling in our quiz. Based on your answers, you are deeply unsuited to any career in the civil service." Which was slightly depressing. It did have the kind of knock-on benefit that it meant I had to go back into the lab and make my experiments work, because my kind of plan B had just crumbled in front of my eyes. And one of the big ironies, of course, is that I am now in the civil service, and hopefully I'm not totally deeply unsuited to it, but who knows. Anyway, so I'd done this project with Ronald. I was enjoying my time in the lab. I was still enjoying research despite the ups and downs, but I was not going to make a sort of future in this particular field. And then I got enormously lucky. And I think one of the things that is often underrated in career progression is luck. And the luck I had was that literally wandering through the lab one day came these 2 individuals, Tone Bookhout and Ferry Hagen. Ferry doesn't normally levitate like that. I don't know what he's doing in that photo. And they came wandering through the lab because they were looking for a collaborator. They both worked on a fungus called Cryptococcus neoformans, which is a yeast-type fungus that you see here. And they were interested in its kind of taxonomy and evolution, but it's a pathogen. This is a fungus that attacks animals, particularly humans. And they had no expertise in looking at pathogenesis, but they were really interested in kind of tag teaming with somebody who did know a bit about that, that they would do the taxonomy, and therefore that kind of collaborator could do some interesting infectious disease stuff. And I basically leapt off my bench a bit like if you've seen Shrek, you know, that donkey goes, choose me, choose me. That was me. So I leapt off saying, choose me, because my project's going nowhere, and I'm game for anything. And so together with Tone and Fairy, we started working on this fungus, and particularly trying to understand how a fungus that normally lives out in trees and in soil has retained or evolved the capability to infect humans and other mammals. And that basically, that chance interaction set the scene for essentially 20 years of research afterwards, because I've spent most of my career since then working in one way or another on this particular problem. So I finished my fellowship, or my fellowship was coming to an end in 2004. So I thought to myself at that stage, right, I've got two choices here. I've done a 3-year postdoc in the Netherlands. To be frank, it hasn't been very successful. I have one second author paper, which is not that great for a prestigious fellowship postdoc. So maybe I should do a second postdoc. But weighed against that was the fact that I didn't really know what I wanted to do. I was also a little bit fed up of working for other people, if I'm honest. Quite like working for myself. And also I was not entirely sure whether I wanted to move another country again, and then another country a few years later. And so I was a bit like, shall I do another second postdoc or not? Or maybe I'll just try my hand and see if I can get a tenure track position somewhere. And in hindsight, that's a bit nuts. I'm not sure I'd ever advise someone to try that. But I did it, and I got quite lucky. And I got in fact, very lucky, because I tell this story a bit as well, because I actually applied for a tenure track position at Oxford, and I was invited for interview, and I went off to Oxford for interview, and the interview was an absolute train wreck. It was abundantly clear by the end of the interview that I was not getting offered that job. But fortunately, because I was back in the UK for that interview, I'd arranged to pop up to Birmingham and see my old supervisor and have a chat. And so I got on the train and trundled up to see her. And she said, you know, how did the interview go? And I said, well, it was a disaster. And she said, well, how convenient, because actually we have a post that's about to come up in Birmingham. And maybe since you're here this afternoon, you could have a kind of informal interview and see if you're a fit for that, which I did. And I was successful with that. And so I had this slightly weird scenario of going back home and telling my then girlfriend, now wife, that I'd gone for a job in Oxford, but got one in Birmingham, and was that okay? Fortunately, it was. And on the back of that, I set up my lab in 2005 in Birmingham, and I have been there ever since. And essentially what we have been working on for all that time in my group is this interaction between the host immune system, particularly the innate immune system, and a variety of opportunistic pathogens, so pathogens that typically only cause disease in immunocompromised hosts. Largely Cryptococcus, that fungus that I just mentioned, but not exclusively. We've done some work on bacteria and other fungal pathogens too. And all of that kind of drive around infectious disease has been focused really around kind of one approach primarily, which is this idea of microscopy. And so particularly in the case of Cryptococcus, for example, what we know is that this is a fungus that's present in the environment. We're all exposed to it all the time. It infects the lungs. Most of us clear that infection without ever getting any symptoms at all. But in unlucky people, particularly those immunocompromised, it can cross to the brain and kill you. And so most of my work in my lab has been looking at this movement from the environment to the lung and from the lung to the brain. And largely speaking, what we have done over all that time is a lot of microscopy. I love microscopy. I think you can learn a lot from it. And so most of my lab has been kind of taking live imaging approaches to that. And we've worked on a whole variety of different things at quite a range of scales, actually. So, for example, this movie is when we looked at co-infection. So these are cells that are co-infected with viral and fungal pathogens. Oh, I've just crashed my computer, I think. That's a bad sign. Oh, I'm hoping you can still hear me. I will just stop that share and come back out and come back in. Oops.
We can still hear you absolutely fine, Robin.
You can still hear me.
Just
That's
the
good.
presentation that froze there.
It certainly did. That was that doesn't often happen. All right. Okay. So where are we? Let me see if I can get back. Maybe I won't try that movie again. uh, let's go to here instead. There we go. Apologies for that. Best laid plans and all that. At least I haven't lost Wi-Fi. Right. And how's that? Can you see that one okay, Poppy?
Yeah, perfect. We're back on it. It's like nothing ever happened.
She says very diplomatically. Brilliant. So yes, anyway, so I spend most of my time in the lab doing these kind of imaging-based approaches for single cells, whole organisms, and looking at these infectious diseases. But actually, the thing that was most fun about all of that, I think, is working with lovely people. I'm sure many of you have the same experience. And so I thought I'd share this little picture, which is now several years old, but the lab, shortly before I took up my other role, actually, who were just a fabulous bunch of people to work with on all sorts of things. So that was my sort of potted history of academia. So the question is then, how did I go from basically hiding in a basement on a microscope and move into this slightly weird, nebulous, pseudo-political role that I have now? It's like many of the things I'm saying today, it's not really a plan particularly. It was more of a kind of series of chance and very positive events. So I saw an advert actually just around Christmas 2019 for this role. I thought it was vaguely interesting. And then I got a call from the headhunters who were running the campaign saying, well, had you seen this advert? Which was quite surprising. I wasn't really expecting that. And to be honest, I said to them, I think you've got the wrong number. I have zero political experience or policy experience. The only thing I'd done really outside of sort of independent research was my role as the institute director at the time, running the academic department. And I have for many years sat on one of the scientific advisory committees that serves government. And so these are independent groups of experts who advise government on specific topics. The one I'm on advises on laboratory use of genetic modification. So that's the one that looks at things like knockout mice and viral vectors and those kind of things. But that's it. I have no sort of policy experience at all, really. But I did have this kind of warm and fuzzy feeling about the Food Standards Agency because it was an organization that I felt was quite scientific, quite important, makes the food that we eat safe, and has a lot of sort of scientists within it. And so I sort of thought, this sounds vaguely interesting. Maybe I'll have the conversation. So I did have the conversation, and look where I ended up. The process to appoint chief scientific advisors, particularly, but science experts in the government generally too, is quite long-winded. And in my case, that turned out to be a real benefit because the week I saw the advert, newspapers ran this headline saying mysterious illness in China causing concern. And by the time I was appointed, we were into the first lockdown of COVID-19. And so it takes a long time. But it also meant that I had plenty of time at each step of the way to kind of consider whether this was right for me and whether I really wanted it and to kind of get to know the role as I developed. And actually, I think that was quite important. I think if it had been a sort of all or nothing single interview, I probably wouldn't have applied because it was that iterative process that helped. So broadly speaking, it looks like this, and this is kind of similar for most of these roles, I think. There was a sort of long listing interview, typical CV, cover letter, and have a long listing interview about you and your fit for the role. If you make it through that, one of the things that was slightly unusual for me, having not done it previously in academia, but very common in industry and government, is psychometric testing, which was quite extensive and actually a fairly prolonged interview with a psychologist, digging into my inner psyche and how I performed under stress and all that kind of stuff. And then there's this process which I think is fairly unique to government, which is bizarre but actually quite effective, what they call fireside chats. So I had a series of conversations with senior people in the department and wider in government that were not actually part of the selection process, but they're designed as a way for you as the applicant to get a feel for the people and the kind of role, and for them, I guess, to get a feel for you as a person rather than an applicant. And so I think I had 2 or 3 of those. There was a fairly extensive media test, a kind of pseudo Jeremy Paxman, for those of you in the UK, kind of grilling when I was presented with a kind of disaster that was looming and had to kind of defend myself on what was a kind of recorded fake sort of BBC interview. And then last of all, a kind of proper interview. Oh, and a real life exercise too, where in this case, it was immediately before the interview. where I was provided with three scientific papers, very different scientific papers. I had to pick one of them and then give a 3-minute presentation to someone pretending to be the Prime Minister, essentially. And so these are all designed to test your ability to kind of work at speed and digest information and kind of distill it to a non-expert audience. And anyway, after all that, I managed to land the job. And I have to say, it has been great. I didn't know what I was getting myself into. I've absolutely loved it. And I would really encourage those of you who are thinking, okay, maybe, I've had a great time in academia, but I'm looking for other things to do in my science. I would absolutely encourage you to look at civil service or government type careers, because it is a real, I think people underestimate how much opportunity there is and how interesting it can be. So in my case, what do I actually do? Well, the system in the UK, and again, it's different in different countries, but in the UK, the system is as follows. Every government department should have a chief scientific advisor, and we have about 20-ish government departments. So there are about 20 departmental CSAs. So I am the one for food, for the Food Standards Agency. And then there's, for example, one for the Ministry of Defence and the Home Office and so on and so forth. The system is designed, I think, in a very clever way, in that all of us as CSAs have two reporting lines. So we are responsible to our own department, so in my case, the Food Standards Agency, via the most senior civil servant in that department, which is either a chief executive or what's called a permanent secretary. But in parallel, you're also reporting into the overall government chief scientific advisor. So that was previously in the UK, Patrick Vallance, and now is Angela McLean. And that is, I think, a very important way of keeping independence and balance in the system, because it means that one department can't suddenly go in a different direction against the will of its scientific advisor. And also that there is this kind of interplay between a very focused bit of science that will deliver something for a particular topic and the overall science kind of ecosystem of the country. And those two individuals then, of course, also then report into to Parliament and government via their particular ministers in the case of the department, and via the Prime Minister in Angela's case. And I think this kind of balance is actually a really underestimated and very important part of this sort of science advice kind of portfolio. So what does that kind of advice look like? Well, in my life, and they vary a little bit from role to role, but broadly speaking, we all have something kind of similar, I think, to this, in that you balance these two sets of responsibilities. So there's the set you have within the department. And so that's about the kind of leadership of the department. It's about setting the direction of travel for science. So, for example, science budget, laboratory investments, training and skills. It's about being sure that government has access to the very best possible science and evidence for policy making. So, for instance, if we're about to launch a new policy on, I don't know, restaurant hygiene, how good is the evidence base? Do we really know what works in terms of keeping restaurants clean? How much might it cost? So on and so forth. All of that evidence will go through me for kind of being sure that it's kind of top quality and not basically not fraudulent or not flaky. And then there's quite a big part about being the face of science. So in terms of external media and comms and those kind of things. And that's the within the department bit. And then in parallel, you balance this kind of broader government portfolio. So as well as being responsible for science in the department, you are part, along with the other 19 or so chief scientific advisors, of making sure the country as a whole has a robust science base. And so that is very, very much about collaboration, about networks, horizon scanning. So what are the exciting developments in science that are coming? What are the risks? What are other countries doing? These kind of things. And actually, I think this external bit in particular is in many ways quite similar to academia. It's a bit like, part of my role is going to conferences and hearing what's happening, for example. Things that we all do in our labs, like journal clubs, kind of happen in a similar way, but just a bit different in the civil service. It's been kind of interesting to see how this all works and how it all kind of boils down. And so I had a look before this talk, actually, I had a quick look back in my diary for the last couple of months to work out, sort of roughly speaking, what proportion of my week I do all these things for. And I would say on average, about half my time is spent on what I'd call data and evidence for policy. So that is people providing briefings and information about things like foodborne disease rates and clusters of infection. I spend a lot of time on social science, actually, which I had no background in previously. So consumer purchasing, who buys what and how, what do people do with their chicken when they get it home to their kitchen? What do people do with their pet food? We're responsible for pet food too. And are there areas where we need to do things about public education or change policy or the law in some particular way? And I spend quite a bit of time on trade and food systems. In the UK, about half the food we eat comes from overseas, and so understanding patterns of trade and vulnerabilities is part of the role too. So that's about half the job. I would say I spend about a quarter of the job on the sort of horizon scanning and capacity building bit. And so that is thinking about developments out in the world of science that are relevant. So one that we're spending a lot of time on at the minute, for example, is cell cultivated products. So that's lab grown meat and artificial dairy and those kind of things. Um, Technology, so that might be technologies like AI. We have big work at the moment on AI for regulation. So can we use artificial intelligence to predict food crime, for example? But also hardware technology. So 3D printing of food is the one that's on my desk at the moment. And there's a lot about skills and capability. So do we have the right labs, the right methods, trained experts, people on PhD programs who will provide the expertise of tomorrow, et cetera, to ensure that government has access to what it will need this year, next year, and 10 years from now. And then the other bit of the job, which is very variable depending on what's happening in the world, there are two bits to, I guess. So one is incidents, and we are responsible for all foodborne incidents. And so I spend either no time at all or a lot of time, depending on what's happening in the world, on incidents every week. So that might be individual incidents, somebody having an acute allergic reaction, going to hospital, and in some tragic cases dying from a food allergy. That is something that we look at. But also wider outbreaks. So you might have seen those in the UK. We had an outbreak associated with salad a couple of months ago that was quite big and tricky. And also some things that you don't necessarily think of as being a big issue, but actually are. So early on in my time, I spent a lot of time on sunflower oil because you might know this, when Russia invaded Ukraine, Ukraine is by far and away the biggest producer of sunflower oil, and it essentially shut down sunflower oil markets overnight, which you might think of as not being a particular problem until you realize that every fish and chip shop in the UK operates on sunflower oil pretty much. And so we had to do a lot of very fast work around that to kind of stop the wheels falling off the UK hospitality sector. And then finally, the last part of the role that is, again, very variable in time, although at the minute it's taking quite a lot of time, is on the media. So as chief scientist, you are generally the face of science for stuff that is either very big and very exciting, like new policies, or very big and very scary, like when things go wrong, when the media want to kind of grill someone. And so I spend quite a bit of my time writing or proofreading or contributing to newspaper articles or interviews, doing radio type stuff, and even occasionally ending up on the breakfast sofa, which is quite an eye-opening experience. And that is basically it. The only thing I wanted to kind of finish with, and then I'm very keen to hear all your questions, is that when I told a few colleagues I was doing this, they said, well, you need to end with some kind of top tips. I was like, I'm not sure I have any top tips. But since I've been told in no uncertain terms that you should have a few top tips, here are my three tips. As I said at the beginning, you should always take advice, especially from middle-aged folk like me, with a sense of caution. So you feel free to ignore all of these. But if I had to give three tips, my three would be this. I would really encourage everyone to do what you want with your career and not what you think you ought to do. I was very lucky when I was growing up. I did not have parents who told me I had to be a doctor or a lawyer or inherit the family farm or any of those things. I had complete freedom. I have always chosen to work on stuff that I'm interested in, not stuff that I thought was going to make me money or any of those other things. And I have to say, if you do that and you enjoy, your job, then actually it's never a painful thing. Whereas I suspect, I've never tried it, but I suspect if you do a job because you think other people think you should do that job, it's nowhere near as rewarding. I would really encourage you, within reason, to basically say yes to everything you're offered. At lots of stages in your career, people will say stuff like, ooh, would you be up for going to California for 3 weeks? Or could you possibly give this, keynote talk for me? Or all sorts of things that you might think to yourself, oh, I'm not sure that's really for me, or maybe they don't mean to invite me as an accident. Still say yes. If you can, without totally stressing yourself out, say yes. Some of my best opportunities in life have been when people have sort of inviting me to stuff I wasn't expecting. My favorite one is when someone invited me to give quite a prestigious conference talk, and I happily accepted. And when I got there, I realized they'd actually invited the wrong person. They thought I was somebody completely different, but it didn't really matter. I was there and I got a podium, and actually turned out to be quite a useful moment. So say yes to everything. And last of all, I would really encourage you to be open to surprises. I would never in a million years have thought that I would end up working for government, and definitely not that I would enjoy it, and I have. Um, Along the way, you will make lots of mistakes. I've certainly made far too many to fill an hour's talk with. But they are usually not the end of the world. You know, unless you are a sort of nuclear missile pilot or something, you're probably not going to cause a global catastrophe with those failures. You know, they'll happen. Sit back, pick yourself up, be philosophical about it. It happens to everybody. If there's something to learn from it, but really don't, you know, kill yourself over some kind of stressed out thing that happens. It's life, and actually you can learn a lot from failures, just from successes. And so I'd really encourage you to think about that. And that is more than enough of me rabbiting on. Thanks so much for listening. I hope there are a few questions, and I look forward to hearing them. I'm going to stop sharing and hand back to you, Bobby.
Thank you so much, Robin. That was a really interesting talk. You're the first person we've had on to speak about any kind of policy role. So I feel like I've learned a lot, and I'm sure lots of people in the comments have. We have had some questions. I'm just going to share a couple more slides to show the other career resources that we have available here at Protein Tech, whilst I let Robin have a quick drink of water and maybe catch your breath, and then we'll come to a Q&A. So oh, that's my face again. There we go. So as I mentioned, Protein Tech have a whole host of career resources available for early career researchers like yourselves. We have lots of blogs, we have on-demand videos created both in-house and from external experts who've come in to give their opinions on how to transition from academia, how to apply for postdoc fellowships, how to start your own business, how to create a LinkedIn profile or an online presence. So there really is an awful lot of ground covered. We have all of our resources available in a early career researcher hub you can find on our website by going to our support tab, or you can scan that QR code there. Everyone who has signed up to one of the webinars this week will have all this sent out to you guys in a follow-up email within the next 5 working days. So don't worry if you can't get that QR code working just now. And as I mentioned, today's talk has been part of a Careers Week webinar series we've hosted. And again, don't worry if you somehow missed this and managed to sign up to just this talk independently. We will be circulating out all of the recordings for this and some of the contact information for our speakers. So if you missed the Q&A but still want to get in touch, a lot of people are still willing to answer any questions you have. And finally, anyone who has signed up to one of our webinars is going to be entered into a raffle to win a premium resume or CV package from alma.me, which is the career surfers offered by Elena Hoffer, who was our guest speaker on Tuesday. And with that, I just want to say a huge thank you again to our speaker, Robin, and to everyone who has attended one of our webinars. If you did find this useful and you want to stay up to date with all of the other events that Protein Tech are hosting, both in person and online, you can follow us on any of our social medias just at ptglab.com. So with that, I will stop sharing my video so we can see, there we go. Is it just our faces again now, I think? And yeah, we've got loads of questions. So I'm going to just jump right in then and start off with, well, first of all, Juliana has said, thank you very much for such a fantastic talk. Do you miss being in a lab environment and doing your own research?
Yeah, thanks, Juliana. The short answer is yes, sometimes. I guess in some senses, I was already missing it. So prior to this role, I was, institute director, and as I guess most of you know, in academic departments, the head of a department doesn't get much time for experiments anyway. So I had already stepped away a bit from the lab. So rather than doing experiments, I think I missed actually is that kind of I guess vicariously doing experiments. People, postdocs, students would come to me from the lab and say, look at this great piece of data. And that was really exciting. That happens less often now, sadly. So I do miss that a bit. But actually, it's turned out to be quite rewarding. I really enjoy it. So, for example, in an incident, in a bizarre way, and suddenly somebody comes up and says, look at this, I've got a matching genotype to this one here. I think the incident started here. It's actually the same feeling, but just in a very different way. So in an ideal world, yeah, I'd have both. But although I do miss it, I think there are other things that kind of give you that same buzz, which is fun. having said that, I mean, it is great. I mean, don't let me diminish it. That feeling when an experiment works, you have fab data, that is fantastic. So yeah, if you're still getting that buzz, stick with it.
Great answer. And yeah, we have this question a lot when people are asking about transitioning out of academia, but I think you sort of hit the nail on the head there when you say even when you stay in academia, unfortunately, the more senior you get, the less lab work you do, and it does become a lot more administrative, really, doesn't it?
Yeah, I can think of just a couple of examples of people who really became professor and still did their own experiments. So with some things, it's easier than that. I think if you're doing kind of mouse experiments, there's absolutely no way you can do that. If you're working kind of yeast genetics, you can do the occasional bit on the end. But the reality I very rapidly learned as well is that actually quite often other people are far better than you. I used to, for quite a while when I became group leader, I used to still do my own experiments. I was definitely the rate limiting step in all the experiments we did in the lab, and realized I was much more efficient to let people who were better at it do the job.
So we had another question, which is when you were a new postdoc in a big lab, what were the skills that enabled you to adapt quickly?
That's a really good question. And actually, well, in some sense, I think I didn't adapt very quickly. And the reason I would say the single reason that was most what's the word, inhibiting in doing that was that I probably wasn't kind of loud and pushy enough. So I'd come from this quite small lab. Everyone knew each other. We all did stuff together. Very English in some senses, although Laura, actually, my boss, was American, but very sort of reserved and, terribly polite. And I went to this enormous lab. And actually, what I realized too late was that, if you wanted, for example, to get time with the boss, you had to be quite pushy about it. You had to say, I need to see you tomorrow. Here is my data. And because I didn't do that, I didn't get access. And in fact, my first year, I think I only met with my supervisor twice in my first year. And at the time I felt really resentful, but actually now I realize it was partly my fault. Like, he would have been happy to meet, but he was just waiting for me to come and say, I need to talk to you. So I think the thing I would say is don't be shy about it, be quite encouraging. And the other thing that is really useful, especially if you're in a new lab in a different country, is to look for like-minded people. I mean, there'll be lots of international postdocs and PhD students from all over. They're a great source of, friendship and socializing, but also insight, right? They'll be in different labs. You can form friends and borrow their pipette or their centrifuge or whatever it is. So I think that network is really important. But yeah, my best piece of advice, I think, is be, nice but vocal.
Great answer again. Thank you. So coming on now then to a question very related to your current position is approximately how many people work within your team at the FSA? What kind of roles are they doing? And do you think a postdoc experience is necessary to get those roles?
Right, great question. So go in reverse order. No, you don't have to be a postdoc. We do have postdocs working in my team. We also have people who've come straight from PhD without doing a postdoc afterwards. And we have people who don't have a PhD at all, who've come from undergrad or master's level. So there's kind of entry points all along the way. In terms of size, it's very variable. So the FSA, which I'm in, the department I'm in, is one of the smallest departments in government. We are in total 1,500 people, of whom about 200-ish are scientists. And actually, my position is this kind of slightly luxurious one that they're either all belong to me or none of them do, depending on how you interpret the role, because I'm formally responsible for all the science in the department. So they all kind of feed into me, but I don't formally line manage any of them. So in principle, they're all independent. So that's kind of, as far as I'm concerned, ideal. I get all the benefit and none of the hassle. But then, yeah, so that's about 200 scientists out of 1,500. For comparison, we do an awful lot with DEFRA, so that's the Department of Environment, Food and Rural Affairs in the UK. And that is about 10 times bigger. So totally DEFRA is about 10 or 15,000 staff. And they have something like 1,500, 2,000 scientists in DEFRA, I think. So it can be quite big. And then you have a bigger hierarchy. But yes, overall in the British civil service, about 10% of the British civil service are scientists. And I think one of the really important things is that many of those are not in science roles anymore. They've transitioned into policy roles or these kind of things, but also the other way around. One of the people who's really instrumental in my team in terms of the science is actually a medieval history graduate. So there's a lot more flexibility.
That's really interesting. So thank you so much. Yeah, I think unfortunately knowing the ins and outs of how the government and civil service positions works, it feels quite isolated until we have someone like yourself who can obviously share that information with us. So I've just seen a question that I think is really interesting by Connor. So Connor has said, thank you for sharing your journey. Could you please share how basic researchers can best interact with policymakers to make meaningful change to our communities?
Brilliant question. Thanks, Connor. Yes. So there are sort of two answers to that, I think. So one thing, so you can absolutely do stuff without ever having to set foot in government or do any of those kind of things at all, because the evidence that we're using, right, that I'm using there is coming from all of you, basically. So publishing papers, having them visible to kind of policy is part of it. And particularly, actually, you can be a bit kind of pushy about it. So if you're working in an area where you think you've made a discovery that has relevance, kind of impact wise, do feel free to send it off to people. So I sometimes get people emailing me, for example, saying, "Do you know what? I've just made a discovery about this new way to detect salmonella. Have a look at my paper," and that's really useful. That is not like I'm not at all sad about that. I'm really, encourage people to do that. So if you're thinking, I've made a discovery, somebody in government should know, let us know. So that's one way. To more formally to kind of get properly involved, there are a couple of things you can do without actually taking a job in the civil service. One is that you can look for positions on scientific advisory committees. So there are 109 scientific advisory committees across government. They all operate on basically the goodwill of experts, which includes all of you listening. So, for example, if you're an expert in genetics or in animal use or in environmental pollution or whatever, there will be a scientific advisory committee that you can think about applying for. It's a competitive process, but great way to get kind of, your expertise into government. And last of all, there are now some really good sort of fellowships that will help you interface between them. So the Royal Society here in the UK runs a policy fellowship that you can kind of use to borrow time out of academia to come into government, often on a part-time basis. So you don't have to abandon your career. You can just try it for a day a week, for example. And many of the UKRI fellowships now have sort of policy parts within them, so you can get kind of quite formally involved while still definitely being kind of 100% academic. So I'd really encourage you to have a look at those. Royal Society policy, if you Google Royal Society and policy, they have a lot of useful tools actually about how to get a bit more involved in that if you're interested.
Great answer and great question, Connor. Thank you very much for that one. So we've also had a couple of questions on sort of imposter syndrome. Somebody's asked how you manage that directly. Somebody has said, did you struggle with stress when you were starting a position that you knew that you didn't have much experience in?
Great question. Thank you very much. So, did I have imposter syndrome? Absolutely, especially in the beginning. So as I mentioned, I started my current job in the middle of the first lockdown. And for those of you based in the UK, you might remember in these days, we were having daily briefings from the Prime Minister, flanked by either or both of the Chief Medical Officer, Chris Whitty, or the Chief Scientist, Patrick Vallance, every day. And that was absolutely grueling for them. And so actually early on when I joined, there was a slight discussion about, well, how can we sustain this? And maybe we should start kind of rotating these things around. And I thought, my goodness, I know nothing about this job. And now people are seriously talking about, briefing the country. Fortunately, that idea never took off. But I absolutely felt kind of out of my depth. Google was my, love for the first 6 months, because I'd be working from home like most of us, where two screens, one screen with my Teams call or Zoom call like this one, other screen with Google, so I could frantically Google what everyone was talking about and try and offer some contribution. I think the nice thing about science is that, we are, by definition, we are a very honest discipline. So actually people are completely up for it if you say, I have no idea what you're talking about. And so I did a lot of that and kind of learned bit by bit. So that was useful useful kind of starter for 10, I think. So imposter syndrome, yeah, absolutely. And sorry, I forgot what the second part of the question was, probably.
It was just about how you managed your stress about starting a
Stress, that's right.
position.
Yeah. So actually it turned out, so it hasn't been, in some sense, it hasn't been so. I mean, academia is quite stressful, right? We've all written, PhDs and papers and given conference talks, and that's quite stressful. So in some senses, it's not more stressful than that. And you're supported by a really good team in government, so that is great. I'm never, even if you're on the kind of breakfast sofa in front of the media, you're not really on your own. There's a comms person behind the camera, kind of supporting you. You've been briefed, so you feel really well supported. The only time that I find it quite stressful now is when you're making decisions where you have to make a decision, you don't have all the evidence, and actually, kind of lives are at stake. And that is still very stressful. And actually, I think it's right that it's stressful. Like, I don't want that to not to be stressful, because you shouldn't reach a position when you don't, I don't lie awake all night. But I think it's quite good that you take those decisions seriously, and don't feel entirely comfortable with them. I'd be worried about someone who did.
Another great answer, thank you. So we've had a few questions around how to start a career in civil service or in policy. Are there any internships available, or do you know of the best way to sort of make that transition?
Yeah, both. So if it depends a bit on where you are kind of career ladder wise, but if you're, for example, just finished degree or PhD, then there are a couple of options. So there are internships. So you can do internships, sometimes actually as part of, if you're on a PhD program that has a professional placement, then you can absolutely come to civil service for that if you can, so send some emails and encourage people. There are formal internship schemes that some government departments run to have people for sort of 3 or 6 months. Then the main route of entry to the civil service as kind of career at that stage is what they call the fast stream. So there's a formalized, centralized process where you can apply to the fast stream under a science ticket. So you say, I'm a scientist, you apply, it's competitive, you get interviewed. If you get into the fast stream, it's a bit like a rotation PhD. You then get placed in different government departments. The big advantage of the fast stream that no one ever sort of says is that it's incredibly competitive. So, odds are against you, but there's a really big market for the people who just miss out. So even if you fail, in inverted commas, there's a huge number of jobs that are open to you within the civil service for having made it that far. So I'd encourage people, if you're thinking about it as a full-on career, look at the fast stream and don't be put off by the numbers, because coming second can actually still be very good.
I didn't know that. That's really good knowledge. I knew how competitive it was. I didn't realize that it's not just all or nothing again. So thanks for sharing. And a bit of a logistics question though for you is it necessary to be a UK citizen to apply for the UK civil service?
No, generally not. There are some roles that are. So Ministry of Defence, some of those require nationality. Broadly speaking, most roles are open to people who are UK, EU, or Commonwealth citizens generally. But actually, also, in fact, this morning I was in a meeting with a colleague who's American. So there are lots of roles for most nations. I think it might be tricky if you were North Korean. That might be a bit tricky at the moment. But generally, most nationalities you can join with. There might be additional hurdles if you're some nationalities around things like security clearance. But yeah, I wouldn't let your passport be a barrier to you. And actually, on the job advert, it will specify if there are kind of nationality restrictions.
Perfect, thank you. So we are unfortunately slightly running out of time, but we've had so many great questions. I just want to squeeze at least one more in, if that's okay with you, Robin.
No problem.
And I think this is a good one to end on. So first of all, thank you for this incredibly inspiring talk. It's given me a lot to reflect on. If you were sitting on a hiring committee for a policy-related role and you've received a healthy academic resume, what types of qualifications or experiences are going to help you stand out?
Great question. Right. And that's a really good point to kind of highlight, is that they're two completely different CVs. So I think the CV you write when you're applying for a postdoc or a tenure track position or whatever, it's all about papers and grants and amazing experimental methods, right? All of which is basically irrelevant to civil service. What they are interested in are your transferable skills. So they are really interested in things like communication skills, leadership, teamwork, etc., etc. So my advice is that if you're submitting a CV, what you do is you pick out. So don't think to yourself, oh, I've never been on an international government panel, so I can't apply. Think to yourself, what skill do I have from my PhD or postdoc that is comparable with that? So for instance, I did my PhD as a collaboration with 3 international labs. That is demonstration of your teamwork skills and your collaborative skills. Try and pick that out in a way that will be kind of concise and make sense. So you might want to say things like, I don't know, I have regulatory experience because I'm an animal license holder in my lab and I help write the project license, so I've got this experience of Home Office legislation. I went to a conference and I gave a keynote talk. And so, I know how to, to an audience, which was not my normal audience. So I know how to relate to people who are from a different discipline. So highlight those things. And the last thing just to say on that, I think, is in the UK system, and I think it's quite similar in other civil services as well, they have these characteristic-based scoring systems. So it will say in the job advert something like, the skills we are particularly looking for in this are, and then it will be really general things like, public communication and data analysis. Make sure you absolutely hit those directly on the head in the cover letter or the CV. So you say, I have skills in data analysis, including bum 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 bum. Don't leave it to them to interpret it. Make sure you pick out those things from the job advert and you kind of regurgitate it with your examples in the letter, so that it's really easy for the selector to go, oh yes, they've ticked those boxes. Let's have a look at them at interview. Because if you don't tick the boxes, it's really hard actually to shortlist someone for interview if you can't demonstrate that they've ticked those boxes.
Great, thank you so much. So I think just because of the time, unfortunately, we are going to have to end it there. But this has been, I think, a really, really useful conversation. We've had lots of great comments and questions about how to get involved in policy. So I think, you've definitely helped a lot of people with the first step in their career there. So again, I just want to say a huge thank you to both yourself, Robin, and to everyone who's come and asked questions today. And I'm going to wish you all a wonderful rest of your day, and I'll bring the webinar to an end. So thank you very much.
Thanks so much, all.